Hello, everyone. So this is the last session of our real-world impact and regulatory decision-making track regarding modeling and simulation in drug development. And for this last session, we're going to have Luiza Borges, which is a specialist in health regulation in the Brazilian regulatory agency, ANVISA. She works pretty much with this therapeutic equivalence department and involved in a lot of activities on modeling and simulation there. So thank you very much, Luiza, for joining us. The floor is yours.
Thank you, Gustavo, and thank you, simulation teams, for inviting me. I will share my screen.
Yes.
Okay. I will present on PBBM applications to support regulatory interactions from the perspective of the Brazilian Health Regulatory Agency, ANVISA. I must start from the disclaimer, so this presentation reflects my views as presenter and should not be construed to represent ANVISA's views or policies. In this presentation, I intend to cover a brief overview of PBBM models, its potential applications both in drug development and regulatory decision-making, some highlights of PBBM cases that were submitted and assessed by ANVISA, and talk about the regulatory scenario, initiatives, and perspectives in Brazil. At this time of the event and for this audience, I can assume that everyone's quite familiar with PBBM, but I must start from the beginning. So the acronym PBBM is being used to physiologically based biopharmaceuticals models, which can be detailed as physiologically based models that are applied to biopharmaceuticals assessments. And as biopharmaceuticals study the relation between the physicochemical properties of a drug and its dosage form and its bioavailability and pharmacokinetic, we see that the most important pharmacokinetic process that should be described mechanistically by a PBBM model is absorption. So necessarily, a PBBM model includes a mechanistic absorption model, which in GastroPlus is the ACAT model. But the other process, the disposition process, can be modeled in simplified ways as compartmental model or minimal PBPK, or even in a full PBPK. So sometimes found in the literature some publications that are PBBM but are as well as PBPK for absorption, PBPK absorption. Here is also important to emphasize that the ultimate goal of biopharmaceuticals studies is to link drug product quality to the drug product in vivo performance, aiming to ensure to the patient a consistent delivery of the therapeutic benefit on a batch-by-batch basis. And this figure from the work of Wu and collaborators greatly depicts what I've explained. So PBBM focus on providing mechanistic understanding of how drug product quality attributes interact with physiology, influence the in vivo performance. So inputs of the model are drug-dependent parameters and formulation-dependent parameters, emphasizing the relation between critical process parameters, material attributes, and quality attributes. The physiological parameters are normally set in the model for the species of interest. We integrate this with a disposition model and then building and validating the model against a relevant clinical data set with the relevant variants of formulation, we are able afterwards to use the model to predict the effect of different dosing regimes on absorption and PK. Then PBBM permits to establish mechanistic VIVR or VIVCs and or to perform virtual bioequivalence studies, being available to connect in vitro results, especially dissolution, to in vivo performance, normally as pharmacokinetic results. And among the biopharmacokinetics toolkit, we can say that PBBM has a unique relevance to perform BE risk assessments, to support biowaivers, and to set patient-centric quality standards, like clinically relevant drug product specifications. We see and recognize PBBM as an evolving tool which can be used throughout drug product development, regulatory approval, and lifecycle management. And here, I try to explain these relations between potential applications in drug development and regulatory decision-making. So for drug development, PBBM can leverage data and be an enabler for quality by design and model-informed drug development incorporation. So being able to enhance the knowledge about quality attributes that may affect absorption and PK, known as critical bioavailability attributes, to population strategy development, and to optimize biopharmaceutical assessment with better design or even avoiding some BE studies. Its application in drug development has a relation with application for regulatory decision-making, but for regulatory purpose, we must bear in mind that the model impact must be balanced, considering the proposal risk level and also the model credibility assessment. So always the decision-making is done in a stepwise manner, considering the whole body of evidence and taking account the risk. Here a list is a non-exhaustive list of potential applications for regulatory interactions. We can develop a VIVR, a VAVC, and safe space to support clinically applications for critical bioavailability attributes like dissolution, API particle size, and percentage of polymorphic forms. Develop a model to support BE risk assessment or even a biowaiver for post-approval CMC and SUPAC changes, for instance, when they failed the in vitro dissolution similarity test, to support for different strength with non-proportional formulation, for BCS class 3 drugs that has non-qualitative and quantitatively the same composition, non-Q1, Q2, to assess food effect on BE, to assess the potential of pH-driven drug-drug interaction, and also to assess absorption and pharmacokinetics in patients with altered GI physiological conditions. Straight a little bit with highlights of cases that were received by ANVISA. Since 2020, we received 6 cases, formal applications that vary in objectives. So 2 for BE risk assessment for CMC post-approval change, 2 for biowaiver of different strength, 2 to support dissolution specifications for register or post-approval. Cases 1 and 2 are both from new molecular entities and for CMC post-approval changes. Case 1 is a major post-approval change for which BE is required. But the issue is that before doing the BE, you must do the dissolution profile comparison and the solution profile comparison with F2 and also Mahalanobis distance. So the proposal of the company was to build a mechanistic VAVC to support a biowaiver. The product was an extended released formulation of a BCS class 3 drug. All these cases I will present had discussions with the companies and the questions raised by the agents, so here's highlight of major assessment points or issues. We see in this case uncertainties in dispositions, mainly due to the lack of IV data. We see the lack of experimental solubility data, and most importantly, the data were not able to show that the QC solution method were biopredictive. So a correlation equation and validation of the proposed correlation were not shown. And after all, this proposal were denied, and a clinical studies were requested both in fast and fed states. Case 2 is similar, but the post-approval CMC change is not major, so B is not required. It could be approved based on dissolution data, but the dissolution data failed when compared using F2, Mahalanobis distance, and also bootstrap. So the company proposition was to develop a PBBM and virtual bioequivalence to support the BE risk assessment. It was for immediate release formulation with a BCS class 4 compound. Major assessment points were the assessment of the precipitation risk, the establishing of a Henke order relationship between the in vitro dissolution and PK, and the supportive IV-AVC, considering relevant, and how variability, both intra-subject and the inter-subject variability, were incorporated in the model. After discussion and refinements, the model were approved. It was considered fit for the purpose. Case 3 and 4 came both from generic industries, and they aim to support a biowaiver of a different strength. So the company had done the bioequivalence study with the reference formulation in one strength and had another strength that were proportional in formulation, but have failed in the dissolution test. And then the company's proposal to develop a PBBM VBE model to support biowaiver. Case 3 came from BCS class 1 or 2 drug with immediate release formulation. And the issues we assessed is that there isn't enough knowledge about product critical quality attributes to support the model. There were uncertainties in the dissolution method and also in the dissolution incorporation, and the lack of the validation of the model considering a clinical relevant and different data set. So the proposal were denied, and the only one strength were registered as generic based on a clinical BE study, and a total evaluation of the quality of the product were requested. Case 4 is for immediate release formulation, BCS class 2 drug. We see uncertainties in dispositions due to the lack of IV data. The API has some specificities considering the PK. It's a highly variable drug that has a dose-dependent food effect. The proposal was to incorporate product dissolution particle size distribution as a critical quality attributes, and you should discuss some uncertainties in its incorporation and estimation. But the company decided to perform the clinical BE study while the model were being assessed and found a different result, so the company withdrew the submission and went to a reformulation process. Case 5 and 6 are from new molecular entities to support the dissolution specifications. Case 5 is to support a post-approval widening of the dissolution specification after out of results having been saw with the QC dissolution method and the specification. So the proposal of the company were to develop a physiologically based dissolution test and use for a PBBM VBE model to support a new dissolution specification that is clinically relevant. The dosage form is immediate release BCS class 1 compound. We discussed with companies the uncertainties in the integration of PBDT data in the model, uncertainties in the relation between the two dissolution methods, the biorelevant and the QC, and the limits proposed, and uncertainties in the variability both within and between subject variability incorporation. After all the discussions and refinements, the regulatory decision was to approve this model as it was fit for its purpose. And case 6 is an interesting case where the company performed a model that used this to justify a risk assessment for the dissolution specification. The company the clinically relevance of the dissolution specification, associating a PBBM model with a safe space and also a PKPD model. It was for immediate release BCS class 2 drug. And despite the fact that this is a very robust and complete model, due to some specificities like the timing of the submission and all the data that was submitted together, for our decision making, this model were considered informative, but we recognize that the model were used for other regulatory purposes and also to support the drug product reformulation process. Now moving to the regulatory scenario, initiatives, and perspectives. All these cases were assessed by our agency and discussed on a case-by-case with the company, mainly through meeting interactions. We recognize the need to have local modeling simulation, PBPK, and PBBM guidelines. But we are currently mapping alignments and amendments that are needed in our legislations in teams that are related to biopharmaceutical analysis, like quality by design, CMC, SUPAC changes, dissolution, IVAVC, BE, and biowaiver. We also foresee the need to have biopharmaceutical risk assessment frameworks in place to help both the industry and the regulators to place the PBBM model according to a risk-based evaluation. So we see some great initiatives published in the literature, like the BioRun roadmap, and also the FDA biopharmaceutical risk categories and decision trees. We are evaluating these tools. We also have decision trees that could be applied to the assessment of some critical PBBM parameters and the steps like solubility, precipitation, integration of the solution, or drug product particle size distribution. And the pillars of the work you have been doing and to do is to build this on the best scientific practices, the regulatory convergence, and also in reliance pathways, as we recognize the limitations, the resource limitations that our agency has to deal with this theme. And we see a great value in regulators, industry, software developers, academia, collaboration initiatives, and all the progress we have made so far are based on this. So I can mention here the training sections we had with GastroPlus in 2020, the invitation to be part of regulators PBBM collaborative study, the working group you have established with experts from local academia to discuss best scientific and regulatory practices. We also want to gain more experience with other biopharmaceutical in vitro studies, with other software platforms, and strengthen our presence and our network in the pharmacometric discussions. I'm glad that the agency will be part of the PBBM best practice for drug quality workshop that will take place in August this year in the United States, and is sponsored by the University of Maryland Center for Excellence in Regulatory Science and Innovation, the FDA. This is a conclusion of the IQ Consortium PBBM collaboration study, and we are glad to be there to discuss the best practice and the ways forward. We also envision to have a workshop framed to Brazilian audience in 2024, when you can discuss the best scientific and regulatory practices, but framing this according to our local issues and discussing, besides PBBM, biopharmaceuticals in vitro studies, quality by design implementation, and have more involvement of other stakeholders in ANVISA, and also involvement of the local generic-based industry and local academia experts. With this, I conclude my presentation. I would like to thank for the attention and let my special acknowledgements to all the team that have been working in this team in ANVISA, and the professors, the reviewers, the simulation team, and also Dr. Greg Hulo for inviting the agents to participate in this collaborative study with the IQ Consortium. Thank you.